So guys, until now you have seen that how we can form or synthesize a glycogen molecule from a primer. A primer is very much required, and once primer is formed, you can elongate the glycogen chain and make the molecule bigger and bigger. But that is, we have yet talked about the glycogen molecule or elongation in glycogen, which is formed because of the alpha 1,4 linkage. That is the linear formation of the linear molecule of glycogen. But we know that glycogen is a branched molecule. Branches है इसमें, हर 10 से 8 molecule residues के बाद इसमें branching आती है. Now question occurs, then how does that branching occur? इस molecule में branching कहाँ से आती है? So that's also very interesting and very, what we say, it needs some focus from you. आपको थोड़ा सा इसको फोकस से ज़रा गौर से सुनना पड़ेगा. These branching are made by the action of the enzyme known as amylo alpha 1,4 to alpha 1,6 transglucosidase. Once again, amylo alpha 1,4 to alpha 1,6 transglucosidase. This enzyme is involved in the branching. What actually happens is that this molecule, it takes, let's say for instance, this is a linear molecule of glycogen made up of alpha 1,4 linkage of the residues of the glycogen, and then it takes some short fragment of it of whatever length, and then it takes it over here and it combined with already growing linear chain of the glycogen, and as a result of this, a alpha 1,6 linkage occurs between these two fragments of the glycogen molecules, and as a result you get the branching. Also keep in mind that one important aspect of the ends of the glycogen molecules, that their ends have a non-reducing sugar moieties, non-reducing glucose moieties, and because they are non-reducing moieties, so they are more facilitating in the formation of 1,6 linkage because of this non-reducing nature. So eventually you will have branched molecule of glycogen, and the enzyme while of course I have already told you it will be alpha 1,4 to alpha 1,6 transglucosidase, and once branching does occur, then again glycogen synthesis again add up the sugar residues in alpha 1,4 linkage pattern and prolong it, elongate the molecule, and then again when branching is needed, the branching occur again through the activity of the transglucosidase enzyme, and eventually you end up with, let's say, 50 to 50,000 a molecule of the size of containing 50 to 50,000 residues of glucose. That shows how huge is the glycogen molecule. There are two type of what we say the molecules of glucose glucose monomers. I mean, when glucose monomers linked with each other give rise to two types of molecules. One is glycogen and one is amylose. Amylose is usually plants in plants, is usually present in plants, while the glycogen is present in the animals. Glycogen highly branched, while the amylose is most of the time linear. Because of high branching of the glycogen, it's very much soluble in water also, and that is why it is readily available to give rise to sugar molecule. And once you have got the branching, the branching pattern, if I show you here, it is something like branches, big, I mean, a huge molecule contains numerous level of branches, as it is shown over here. You can see. So you can see this is linear molecule, then branching occurs, then it goes, and then branching occurs, then it goes, and then branching, then it goes, and then branching occurs. And in this way, linear molecule, then branching occurs, then linear, and then branching. And at the end of the day, you get a very a molecule of an extreme size, which have many branches. And this pattern of branches and this size help in its storage capability and its ability to provide glucose whenever we need.